Hello friends, this video on Biomolecules Part 15 is brought to you by examfear.com. No more fear from exam. Talking about the function of globular proteins, they play a very important role in metabolic reactions. Some of the examples of globular proteins are enzymes and hemoglobin. Now we will have a detailed discussion on enzymes and we will see that they play a key role in the metabolic activities or metabolic reactions taking place in our body. So hemoglobin, enzymes, they all are globular proteins. So these proteins mostly have complex tertiary or quaternary structures. So what are these structures? So we will now talk about the structure of proteins. Now proteins have four types of structures are observed in proteins: primary structure, secondary structure, tertiary structure, and quaternary structures. So these four structures are seen in proteins. So look at this complex structure. So when you look at this complex structure and if you try to magnify a small portion of it, a small portion like this, if you look at the base, what forms the skeleton of the structure of protein? It is made up of amino acids. So the sequence of amino acid is the primary structure, which you can see here. This sequence of amino acid forms the backbone. So linear sequence of amino acids in a polypeptide is the primary structure of a protein. So over the primary structure, secondary structure is there, which takes place by further folding. Tertiary structure happens on even further folding. So that is how we proceed to different structures one by one, which are more complex. So this primary structure or this sequence of amino acids is determined by the genes and the genetic hereditary material DNA. This correct sequence is very, very important for the function. In fact, each type of protein, what they function, what is their role, that all depends on the sequence of amino acids. That is, primary structure defines the protein. It defines the type of protein it is. So that is the primary structure. Let us look at the secondary structure. So now if you look at some of the structures in this complex structure, you will see some coiled structures like this, and you will also see some plated structures like this. So these are secondary structures. So primary structure does not make a protein functional. It just gives the basic shape, or it just makes the backbone. So it further gets coiled to give a three-dimensional structure to the protein, because a three-dimensional structure is needed for the protein to be functional. So how this three-dimensional structure comes into picture? By interactions with immediate neighbors. So what happens, let us suppose if you have a long chain of polypeptides. So all these are, say, molecules, they are all neighbors, carbon, oxygen, nitrogen. So now if they start interacting with each other, what will happen? Maybe they both will try to come near, again they both will try to come near. So folding will happen. So foldings will take place. So this folded structure is the secondary structure. Now, two types of structures are observed in secondary structure. Sometimes it is seen that they gets folded in a pleated structure, or sometimes they are a coiled structure. So this is the coiled structure and this is the pleated structure. So the coiled structure is nothing but the helical structure or helix. This shape is known as helix. So this type of structure is the helical structure. So here if you see here, this gives a better explanation. Here you have the hydrogen bonds. This bonds denotes the interaction between the neighbors, these hydrogen bonds. And because of this interaction, they are together. So this secondary structure is caused by intermolecular and intramolecular hydrogen bonding. Now, each of the amino acid of a polypeptide will interact with its immediate neighbors, forming hydrogen bonds. This will cause folding in the structure. So it gets folded into coils. So this coiled-like structure is the secondary structure, which is helical in shape. There is another secondary structure which is in the form of pleats. When I say pleats, you remember the pleats of your skirt. The skirt which you wear, it has pleats, right? So in a similar fashion, due to interaction between neighbors, you have these kind of structures due to intermolecular hydrogen bond. So this type of structure also gives the appearance of pleats. So that is why it is called pleated structure. However, this pleated structure is comparatively less stable when compared to the helix structure. So in secondary structure, you have two types of structures, helix structure and pleated structure, and pleated structure is less stable than the helix structure. So this is about the secondary structure. Let us look at the tertiary structure. So in tertiary structure, it gets even more coiled and even more folded. So here you can see this has been magnified here to show how it happens. So here bonds between neighboring amino acids results in further folded structure. So earlier the first one was only a sequence of amino acids, so no interaction with any neighbor. Secondary structure came due to interaction between neighboring polypeptides. Right? Now the tertiary structure is coming due to interaction between neighboring amino acids. So it is not only the polypeptides which are interacting, it is also the amino acid as a whole. Like amino acid is what? Amino acid is made up of many peptide bonds, right? So those peptide bonds are interacting with each other. Now we see the amino acid as a whole, this whole unit as such is interacting with another amino acid. So because of this interaction, so please understand this fact. Let us suppose this is one amino acid. This is another amino acid. So all these amino acids together are forming the protein. Inside this amino acid, so you have many bonds inside them. You have many bonds inside them. So what is peptide bond? The bond which is joining the two amino acids, right? Similarly, you have another bond which is joining this amino acid to another amino acid. So it says that these bonds will be interacting with each other. So due to the interaction or between the different polypeptides, secondary structure will come into picture. Now when folding will happen, then these amino acids will also start interacting with each other. So due to the interaction between these neighboring amino acids, there will be further fold in the structure. So the interaction between the neighboring amino acids will make the structure even more folded and coiled, and that will give rise to the tertiary structure. So here the helical structure gets further folded or twisted due to which kind of force? Now which kind of force, with which kind of forces this amino acids will interact? Mostly by weak forces like van der Waals force or hydrogen bonds or sulfide bonds or electrostatic forces. So with all these forces they will start interacting, and based on that the structure becomes even more coiled. Now some of these tertiary structures, if you look at this highly coiled structures, some of them are spherical in shape and they are called globular proteins. Some of them form long fibers and they are called fibrous proteins. Now it depends, this tertiary structure also depends on the primary structure, the type of amino acid present, the number of amino acids, the sequence of amino acid, because here everything is happening due to interaction between the amino acids. And to know the type, sequence, number of amino acid, the primary structure is of help. So tertiary structure depends a lot on the primary structure. So this is the tertiary structure, and mostly wherever you see pictures of proteins, they are generally tertiary structures, which look quite complex. So as I said, tertiary structure depends upon the primary structure. Creates active sites for enzymatic proteins. Now proteins, there are many proteins which act as enzymes. Now these enzymes have got specific sites where the reactants can join them. Now we'll learn about active sites when we talk about enzymes. So for now you just understand that it is the tertiary structure which will create those active sites for enzymes. Now when we will talk about enzymes and their active sites, that time we will discuss this part in detail. So now the last and the final structure, that is the quaternary structure. So here multiple chains of polypeptides are held together by hydrogen bonds. So here now you have multiple chains itself. So let us have a quick comparison between what we have studied. When we talked about the primary structure, we were talking about just a sequence of amino acids. That's it. So we were just talking about one simple chain with the sequence of amino acids. When we spoke about the secondary structure, we said that this straight chain gets coiled due to interaction between the polypeptides in it. When we talk about the tertiary structure, we said that it is not only the polypeptides which interact, it is also the amino acids which start interacting with each other. So it gets further coiled and folded somewhat like this. Now when we talk about the quaternary structure, we mean that we do, so till now, till tertiary structure, we just had one chain. The chain is one, it is just that single chain is getting coiled. So this entire thing is just one chain which is getting coiled in this fashion. But when we talk about quaternary, we mean to say that we have one chain like this, we have another chain like this, and both these chains are interacting. So the structure is becoming all the more complex. So that is our quaternary structure. So now we have two different chains which are held together by hydrogen bonds. So when we talk about the structure of proteins, we see that all the protein structures are highly dependent on hydrogen bonds. So this type of structure is not seen in all proteins. It is seen only in proteins which has multiple chains of polypeptide, because if you have only one chain of polypeptide, tertiary structure is the maximum that you can get out of it, because quaternary structure is possible only when hydrogen bond binds two different chains of polypeptides. So that is your quaternary structure. So let us have a thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.